Hi, everyone. My name's Jeremy, as Amir said, and you got my last name right, which is very unusual. Well done there. So my role at EMSL is to help users apply NanoSIMS analysis to their projects. That's basically what I do. So we'll work everywhere from experimental design, sample preparation. I'll do the analysis for you, probably process the data and try and help you understand what the hell I've given you, which is sometimes a bit of a challenge. So we're a bit of a one-stop shop for the NanoSIMS analysis here, so that's kind of good. So today I'm just going to give you a quick introduction on what NanoSIMS is and isn't, because it's a question that we sort of that I see a lot when I sit in user meetings. So I thought I'd try and dispel some of the myths, and then just go through a couple of applications that we've used it on in plant systems. So the name NanoSIMS is actually just the manufacturer's name for the machine that does nanoscale secondary ion mass spectrometry. So what exactly does it measure, you ask? Well, put simply, it just measures ions, which are just charged atoms, be they monoatomic or small polyatomic atoms, negative or positively charged. It's a destructive surface analysis. So destructive surface analysis. So we have a primary beam, in this case cesium, which is hitting the surface of samples, ejecting a heap of secondary ions, neutrals, whatnot, and we're collecting those secondary ions, sorting them and measuring them. So we're just basically counting the ions. So I did have a really cool video here of how it looks, but it doesn't seem to want to work today. So we'll just skip over that part. But it's important to know that NanoSIMS is kind of unique because it can work across the entire periodic table, all the way from hydrogen, all the way through to plutonium and everything in between and beyond that. So there's lots of potential there. For biological stuff, we tend to work on the lower side and the organic type elements. And finally, an important feature of NanoSIMS is that it has a capability for imaging. So we can raster this primary ion beam over a sample, collecting ion species along the way, join up that data, to create a beautiful image at the end. So the example I give here on screen to show what sort of data we do collect, obviously this has nothing to do with plants, this is a pyrite grain, but it does explain what the potential of NanoSIMS is and how it could be applied. So on the left of screen here, you can see we can target elemental features. So in this pyrite grain, we're simultaneously collecting ions of a bunch of different things that you might be interested in. Here, we're looking at nitrogen, arsenic, oxygen, copper, but that's interchangeable. We can select whatever we want with that. So this is all one analysis. It's all simultaneously collected. We can overlay these elemental features and come up with something like this to get an idea of the relationship between those species we're interested in. I think what's even more interesting and kind of cool with NanoSIMS, we can start to look at isotopic features. So in this example here, we've looked at sulfur isotopes. So we have the rare 34 sulfur isotope, rare 32. We simultaneously collect images for both of those guys, and we can overlay those to create a ratio image. In this example, it's a pyrite grain, so this is a natural abundance, so we can see nice isotope fractionation, and that probably means something to the geologists. But you can see how that could be easily manipulated in biological systems by adding isotope traces and looking for specific metabolic processes in that regard. So the other feature here is to do with resolution, and this is another attractive feature of NanoSIMS analysis, because we've got a resolution on these images typically between 50 and 100 nanometers, but we can actually push that down to 30 nanometers if we'd like. And the next generation of machine is reported to be close to 10 nanometers resolution, which would be kind of interesting to see. So on the schematic on screen here, I try to give you an overview of how the NanoSIMS works. And I think once you understand this, everything else makes perfect sense what I'm talking about. So the NanoSIMS is basically a primary ion source hitting the sample. We can raster that across if we'd like to create the image. And it's ejecting secondary ions, along the way. And these are counted, sorted based upon mass charge, magnetic sector, and this flight tube here, where we have the lighter ions down one end, heavier ions up the other. And we're physically moving detectors, collect the ions that we're interested in. So an example here on the right of screen, where if we would like to use detectors 1 and 2, for example, to collect mass 12 for 12 carbon and mass 13 for 13 carbon, so that we could generate that 13 carbon isotope map. We would run a scan along, we'd run a scan to find out the exact radius of the magnet where we want to be. In this case here, we want to be about here. So we would physically move this detector on really train tracks on a small scale. And that's where that detector is going to collect those 12 carbon. It's a little trickier for 13 carbon. Again, same thing, we're going to collect the ions that we're interested in, but here we have an interference of another ion species at mass 13. So 13 carbon and 12 carbon and hydrogen, the mass is almost identical. I think there's a difference down on the third decimal place, AMUs, but NanoSIMS has the capacity to separate out, or has the mass resolving power to separate those peaks out, so we can be pretty confident we're collecting the 13 carbon. On top of those two ions we're collecting, we can simultaneously collect a bunch of other things that we might be interested in: nitrogen, sulfur, chlorine. You know, the world is your oyster on this, whatever you might be interested in. But we can strategically do that as well. For example, nitrogen is always a great one to look at biological features, biological structures of the sample. So here we've got a bunch of yeast cells, and you can start seeing the features there without having to go into an electron micrograph or optical image. So once we have this 13 carbon, 12 carbon detectors worked out, like I said, we can overlay those to create a 13 carbon enrichment image, and then we can go into those images and select regions, pull out the counts for those particular pixels, and then we come up with this answer for how enriched that particular yeast cell is. And based on counting statistics, get an error associated with that and work out whether it's significant. So what I find kind of overwhelming for users in this whole field is just how many options they have for doing this type of work, even within a sort of a niche field like imaging mass spec. There's just so many different things. This great image here shows you all the different modalities you could possibly choose. And even over here, you can start to see the choices we have with the sensitivity and resolution potential there. But it's kind of not the whole story. I think each one of these techniques has its own pros and cons. And if you look at something like, obviously I'm talking about NanoSIMS, so comparing it to something similar like TOF-SIMS, it looks pretty similar up here, but realistically, it's going to give you a whole heap of different data out there. So TOF-SIMS is going to give you that lovely spectrum where you're getting a bunch of information for heaps of different things. NanoSIMS, on the other hand, is very targeted and a little bit better sensitivity, and we can start to interrogate that isotope idea a little bit more and apply that and manipulate that to our samples. So I'll jump along to a couple of examples here of how we've applied NanoSIMS in the plant sciences. This is from a user project here. This is work done on the last few weeks, so it's relatively fresh. I'll just give you a simple rundown of the experiment. You don't need the details here. The project was basically looking at nutrient uptake in Brachypodium. I know this is a Arabidopsis, but I couldn't find a Brachypodium late last night. That's Brachypodium. So the users actually basically had these plants growing on Petri dish and applied the nitrate solution, left it for, I think this was for 2 hours or something like that. We harvested the roots, collected the roots, fixed them, dehydrated, resin embedded, sectioned them down to about 200 nanometers, sort of electron microscope sort of sectioning deal, and then imaged it on the NanoSIMS, specifically looking for the accumulation of this 15 nitrogen label. Now, I wanted to have a look at the entire root system, which is which is pretty difficult on NanoSIMS because generally we work at this kind of 45 micron square scale. Plant root is a hell of a lot bigger than that. So anyway, we persevered, and the idea here is that I was able to stitch, I think there's 120 images here together, generate this entire section. It takes a while to do. But I thought it was important to do this because I wasn't just interested in where the 15 nitrogen was accumulating, but I think it's important to know where it's not accumulating types of systems. And we learned just as much from that in that respect. So this root section here, it's obviously a cross section. See some of the root hairs here on the outside here. It's after 2 hours of the 15 nitrate. And it's not particularly clear here. We'll zoom in a second. But the accumulation of the 15 nitrogen was evident in the steel of the plant, like you'd expect. So what we're looking at here, this is a 15 nitrogen concentration map. Black is areas where there is no nitrogen. Blue is areas where it's natural abundance nitrogen, just run of the mill every day, nitrogen concentrations. Anything that's sort of this orangey, greeny, pink here represents where the accumulation is happening. So we zoom in a little bit closer and we start to see that, as you'd expect, the metaxylem features, these ones here, 1, 2, 3, 4. That's where the accumulation is. Metaxylem obviously being sort of that pipe work where the water's traveling up to the foliage of the plants. So there's a couple of things that are pretty interesting with this data. The first one is for the isotope savvy people out here, you'll notice that this is really subtly enriched. Generally, when people do these types of experiments, they want, a big number on this before this decimal place here. They want 10%, 15%, to be sure. But what we've been working at quite a bit here at EMSL is working down on that small scale, looking for really subtle shifts. So we don't have to bombard the samples with unrealistic amounts of isotopes to answer our questions. And I think this is a perfect example here where we see exactly where the isotope is. It's enriched. It's not very enriched, but it's significantly enriched, and we're pretty confident about that. The other thing with this was kind of cool. You can see here we've got metaxylem here, enriched, but the same metaxylem on the other side of the root are not enriched. And this is actually a feature of the sampling strategy here. When the guys put it in, they only added the 15 nitrogen tracer on one side of the root. So that was kind of interesting and something we've never seen, thought to take advantage of. So I'll go to the second example here, and we'll skip over to Norway, into the boreal forests. So the boreal forests are the biggest terrestrial biome on Earth. But what's interesting about them is that the productivity in these systems kind of defies the nitrogen limitation that's also occurring there. And it's always been a bit of a question mark why that's occurring. So one solution was that there's abundant moss growing these guys, and in that moss there's heaps of cyanobacteria, and they're all fixing nitrogen. Theory is nitrogen's fixed by the cyanos, passed onto the moss. That moss dies after X amount of time, and the other plants in the system are able to take it up, or it's able to cycle through various systems. But what we found is when we did this experiment in situ, we got these growth chambers here where we were labeling them, the moss with 15 nitrogen atmospheric gas, looking for that nitrogen fixation by the cyanobacteria. We collected moss shoots after a while. I think it was 2 weeks maybe incubation. We cut those guys, fixed again, section. So here you have a optical image of the sections. You got the moss stem. These are the little leaves on the outside. Within these little leaves, they kind of form these little pools or wells. These were just teeming with life. Cyanobacteria, bacteria, fungi, algae, you name it, it was there. So we took these sections, we imaged them with TEM to look for ultrastructure to try and identify some of these features. So you can see things like fungal hyphae, green algae, and the cyanobacteria. We then did the NanoSIMS analysis looking for the 15 nitrogen that we'd applied up here as gaseous form. And start to see it in the cyanobacteria here, which is as we would expect. The theory was then that this would pass it on to the moss in some serious mystical manner. Not exactly sure about. But what we found is that the cyanobacteria was in fact fixing the nitrogen, but then it seemed to be transferring this nitrogen out into some of the other microbiota, so into the bacterial cells here, so anything above blue basically. And even down into things like the fungal hyphae that, but it's an interesting way. So what this tells us is that the nitrogen cycling in this system was a little bit more sophisticated than first thought, and there were a lot more players involved, and it was going to be a lot faster than waiting for that moss to die and let that nitrogen enter the system. So the third example is we're going to go from the Arctic down to the deserts in Australia, and this is a bit of a left field one, and one of the original uses of NanoSIMS in plant sciences. It's nearly 20 years ago now, which is insane. This is part of my PhD project, and this involved conservation biology, totally different. Usually you think agriculture in this. But I was working with this particular orchid species here. This is called the underground orchid. It's probably the rarest plant on Earth. There's probably 3, 4, maybe 10 of these plants left in the wild. And it's fully subterranean. It grows and flowers and does everything underground. It's the only plant in the world that actually does that. So it's a bit of a freak, and it's persisted for a long time. It's sort of that ancient relic that somehow has managed to survive in these desert environments in Western Australia, where I'm from. So because it's subterranean, it can't photosynthesize. So how on earth does it get carbon? Well, the simple answer is that very simple answer is that it's eating fungi. So this is a cross section of the roots. All these little balls here, that's fungi growing in there. This orchid's pretty smart. It manages to grow up this fungi, and then it'll lyse these fungal balls after a couple of weeks and basically take up all those nutrients. It's a pretty effective way of facilitating its nutrition. But what's interesting here, okay, it's relying on this fungi, but this habitat here isn't particularly what you'd call a high organic load, high moisture environment that you usually consider orchids growing in. It's a desert. There's no organic matter in the soil. It's like working at the beach, to be honest. So it's really crappy. So there's something else going on there. We also know that this plant here always exists under these shrubs. So the idea was that it's actually forming a tripartite relationship where a plant and plant are connected by this common fungus. And this plant here, the autotrophic shrub, is photosynthesizing like regular plants. Carbon's coming in, transferring it over to the fungus, and then the orchid's able to take it up. That's all well and good, but no one could actually say that for sure that that's what's happening. So we replicated this situation in the lab using a split pot system where we were able to separate the fungal compartments, orchid compartments, and shrub compartment. We labeled it with 13 carbon dioxide and hope like crazy that we'd see it end up in the orchids guys down here. So after this had happened, we'd left it for a while. Again, we fixed the samples, sectioned the samples like we usually do. And we get these sections like this. These little black dots here are those cells just teeming with fungal hyphae. NanoSIMS analysis showed us these hyphae structures here, heavily enriched in 13 carbon. This was the first time NanoSIMS was really used in plant systems. So it's, the data's pretty good. And we'd go back and do it so much better. Good. But I think it was, what it did say is technically it was a feasible idea. It was a crazy system to try on, but it was feasible. So that's a tip. Conservation-wise, it's also really powerful too. Yep. It was a powerful tool for conservation purposes because it showed us that to conserve this really rare plant, we need to conserve the fungi and that other autotrophic shrub and that sort of intricate balance of the nutrient cycling that's going on there. The last slide, what's the future of NanoSIMS analysis look like? I'm going to be really quick here. Essentially, we're a fancy mapping technique. You're going to put some isotope in and we're going to tell you where it is at a pretty high resolution. The beauty is you can go in with other methods, and some of these other methods we talked about EMSL this week, today, sorry, and we start value adding to that. So that's kind of interesting. That's something we're continually exploring. Better resolution is on its way. The new instruments that are coming out of the factory in France are going to be equipped with a much better source that's going to give us 10 nanometer resolution. Whether that's worthwhile, I don't really know. 50 nanometers is good anyway. The big advance is really in this cryo stage that's going to become available for the system. Sample preparation is the big stumbling block here. And if we can really overcome that and create these cryo-based methods where it's a more realistic version of what's going on, I think there's a lot of interesting applications there, not just in the medical world, which big bucks, but for the kind of projects that we're working on. Well, so I'll leave it at that.
Thanks, Jeremy. Nice talk. Any questions?
thank you. Seems to be a great technique. I was wondering how well does it work to resolve rare earth elements, particular differences between them, because I've used other methods, microprobe before, and there's where our problems wasn't differentiating rare earth elements.
No problems at all. I've been working on rare earths in the last month. Featured hybridization probes with rare earths attached instead of fluorescent probes, and it works quite well. And we can use these in concert, so using the bunch together. We just have to be careful. Yeah, that's right. Interferences is one of our problems at the base. We just got to think it through and make sure we know what the potential interferences are, and work with that, because we do see it from time to time. But there's quite a few rare earths.
Right. I mean, I was thinking about natural samples, right, instead of using concentrated to label.
Right. So we're ultra sensitive as well, so we can pick rare earths up, but we quite routinely do. I've worked a lot on geological systems in the past, and that's their bread and butter. And realistically, that's what this method was developed for: geology and cosmochemistry. So yeah, practically every analysis they do is using one of those weird elements of the periodic table they've not heard of.
Thank you. And could you please tell us what kind of underground systems does this orchid live in?
Where?
Yeah, what kind of? It's a cave, or it creates its own hole in the ground, or how?
Just in the dirt, underneath these little shrubs in the middle of nowhere in Western Australia.
Still on the surface. Right?
Still on the surface, so just under the surface, talking 2 or 3 centimeters. Yeah.
That's a super cool plant. Thank you.